Here we go. Okay, today we're going to talk about macromolecules. And in general biology, in Bio 1, when we talk about macromolecules, I'm typically talking about these four basic macromolecules: carbohydrates, lipids, proteins, and nucleic acids. Carbohydrates typically will have a carbon, hydrogen, oxygen ratio of 1 to 2 to 1. For every one carbon, there's going to be 2 hydrogens and 1 oxygen. There's a couple of tiny exceptions when they form rings and structures of that sort, but for the most part, you'll be able to tell carbohydrates from that 1 to 2 to 1 ratio. Lipids, on the other hand, are going to be almost entirely carbon and hydrogen. Once again, there'll be a couple of minor exceptions or additions to the carbon and hydrogen, but for the most part, you're going to see lots of carbon and hydrogen. Proteins are going to have an amino group and a carboxylic group, and also usually some other form of what we call an R group, which means various things could be attached to that point. And then nucleic acids, we'll go into more detail on these when we get to DNA much later on, but nucleic acids are going to have 3 components to them: a nitrogenous base, a phosphate group, and a sugar. We'll start talking about carbohydrates first, and we'll talk about the smallest of the carbohydrates that we call monosaccharides. An example of that would be glucose, and you can see a glucose molecule here, and you can also see a glucose molecule here in what we call a ring structure. So those two really are the same molecule, and if you count them up, you will find that for the most part they have very close to, or exactly, a 1 to 2 to 1 carbon, hydrogen, oxygen ratio. Next in the carbohydrates, we have disaccharides, and disaccharides occur when we take 2 monosaccharides, such as glucose and fructose, and we hook them together. And we'll see this reaction a lot where you hook 2 molecules together. One of the ways we can do this, it's called dehydration synthesis, where we have an enzyme that basically removes the water between 2 molecules and then ends up hooking them together. And then likewise, going the other direction, we will have a reaction called hydrolysis, where we'll essentially add a water molecule between 2 and split them apart. These are all controlled by enzymes, and we'll see them often. So this is the first place I see it, so I'll mention it now. Next in the carbohydrates, we have the very large polysaccharides. Polysaccharides are made up of a whole bunch of monosaccharides hooked together. If we were to break the monosaccharides down, once again using that reaction called hydrolysis where we break it down, we would get a bunch of monosaccharides. One of the things that we'll see is a lot of molecules are built upon some repeating subunit of something. And so if we have a small subunit, we call that a monomer. And if we hook a bunch of that same monomer together, we get a polymer. So some polysaccharides are made up of repeating units of a monomer, such as glucose, and that forms then a larger polysaccharide. Examples of polysaccharides include starch, which are found in plants and things like potatoes and pasta. Glycogen, which is a carbohydrate that we have in humans and other animals that's used to store carbohydrate energy. And then cellulose. Cellulose is a polysaccharide that is typically found in the cell wall of plants. Our next group are called lipids, and there are a bunch of different types of lipids. One of the types of lipids that you'll be familiar with are called fats, and fats are made up of a combination of 2 molecules, one of them being this molecule called glycerol, and the other being this molecule that we call fatty acids. And typically there are 3 chains of these fatty acids, and when you hook them together, you get the entire fat, or in this case, a triglyceride. And so we'll see these as we discuss them more as well. Okay. Next we have in the same lipid category, talking about fats. Fats can be saturated or they can be unsaturated fats. These are also terms you've probably heard before. When we talk about a saturated fat is a fat in which there are no double bonds in the carbon chain. Whenever you have a double bond, it tends to bend the molecule a little bit. So if you look at this unsaturated fat, you can see the carbon double bond right here, and that leads to a sort of bending of the carbon chain there. And so we have saturated fats and unsaturated fats. Traditionally, long ago, it was thought that all fats are bad for you. And then more recently, it became sort of known that unsaturated fats are actually good for you and saturated fats are bad for you. And even more recently now, we're sort of analyzing the fact that some saturated fats that occur in nature might be very good for you, actually. But the problem is most saturated fats, many of them are altered fats that don't exist in nature, and we've added hydrogen to them to make them last longer and things of that sort. And so a lot of food that has high saturated fat is usually high in some form of preservative, and those types of fats end up being not so good for you. But either way, a saturated fat and unsaturated fat, it comes down to the presence of double bonds or the lack of double bonds, as you see there. Also in the lipid category, we have molecules that we call steroids. And once again, steroids, the term steroids is probably something you heard before. Cholesterol is a type of steroid. All the steroids have this sort of basic structure you see here where they have these rings. And so this is what cholesterol looks like. You probably also heard the term LDL and HDL. LDL is a low density lipid. HDL is a high density lipid. And we often call these the good cholesterol and the bad cholesterol, but in reality there's only one kind of cholesterol, and it looks like this. And what we're really referring to is how your body transports that cholesterol through your blood. And when it transports it in the form of what we call low density lipids, it tends to be relatively low in protein, and the high density lipids tend to have more protein as you transport them. And the high density lipid form of transporting cholesterol, that level is considered better for your health than having a high LDL level. But that's cholesterol. There, I should also point out that when you see these ring structures, one thing that's maybe not immediately obvious, in order to draw these and save some time, there's some simple rules. And that is, anytime you have a ring or anytime you have a bend, it is sort of assumed in organic chemistry and in general bio, like we're doing here, that there is a carbon connected at that corner, and that there are, if they don't tell you otherwise, there are two hydrogen atoms connected then to that carbon. So in this particular case, although you only see a couple of H's and an oxygen and an H there on the other end, at the corner of each of those rings that you see there are carbon atoms that just aren't shown, and it's easier to draw that way. That's what the ring structure means. If they're going to put something different there, like an oxygen, they will have to let you know that by putting an oxygen atom there. So that is a shorter notation when you see these molecules that makes it a little bit easier and faster to draw them out like that. Also in the steroid category, we have testosterone. You'll notice that cholesterol and testosterone look quite similar to one another, as does estrogen. So these steroids, these lipid steroids, are all quite similar in their very basic structures with those four rings and some modifications of those. Another type of lipid that we have that is not a steroid, but in this particular case is called a phospholipid. A phospholipid is going to be an important lipid when we talk about cell membranes. It has two important components to a phospholipid. One is the phosphate group, and you can see that here. We call that the phosphate head quite often. It tends to have lots of electrical charges on it, so it binds to water very well. We'll talk about that later on. And then at the bottom, you have what are called the hydrophobic tails, and that is the lipid part of the molecule, and those are usually a saturated fatty acid or an unsaturated fatty acid, and there's usually two of them attached to that phosphate head. So that's what it looks like, phosphate head, and then the what we call the tails quite often are the lipid part of the molecule. Okay, Next group is we have proteins, and proteins we'll go into more detail on as we talk about how they function. Basically, proteins are made out of smaller monomers, small subunits called amino acids, and amino acids are molecules that have a carboxylic group and an amino group, and you can see that here and here. And the way we take two amino acids and hook them together, we once again use that reaction of dehydration synthesis. We remove the water out between them, and we form what's called a peptide bond between the nitrogen group and the carboxylic group of two adjacent amino acids, and we hook them together. And we'll go into more detail on that when we talk about protein synthesis and that sort of stuff. And proteins are very structured molecules, and there are 4 levels to the structure. We start off with what's called the primary structure, which is the polypeptide chain, or just a chain of amino acids. And then what happens is those amino acids will then, based on whether they're positive, negative, or neutrally charged, they will fold and they will form these smaller sort of repeating patterns. Sometimes we call this an alpha helix, or a pleated sheet based on this sort of repeating pattern of folding that they have. And then within that pattern, the tertiary structure is that pattern continues to repeat. And so you might have a couple of alpha helix folds, and then a pleated sheet fold, and then that repeated segment can continue over and over again. And then if you take that as a final tertiary structure protein, and or tertiary structure single chain, and then you hook that to several different groups, you can get a full functioning protein. That's the quaternary structure. That's when you take, say, several of the tertiary structures and hook them together. That's what you're seeing in this case with the protein called hemoglobin, which is used to transport oxygen throughout your body. Proteins can come in different forms. We'll talk about them primarily in the form of being enzymes, and enzymes are proteins that will speed up chemical reactions. We have enzymes that break things down, and we have enzymes that put things together. Kind of an easy way to think about it in the beginning is to think about, say, an enzyme that you typically have in digestion called lactase. Lactase is an enzyme that will digest the milk sugar called lactose, and so that will break that down. And so in that particular case, the enzyme's lactase, and the substrate that the lactase operates on is lactose, and the lactose then would bind to the active site and break that down. In addition to enzymes, we also have structural proteins, and examples of those might be like collagen that are used throughout your bodies, and tendons and ligaments and things like that, and also keratin, which is a major component of your hair and your fingernails. The final group of macromolecules are called nucleic acids, and once again, like proteins in particular, we'll do these much later in great detail later on. But for now, just to sort of define that group, macromolecules that are nucleic acids have these 3 basic components. They have a phosphate group, they have what we call a nitrogenous base, and there's some form of sugar. And what nucleic acids are used for are making molecules such as RNA or DNA. And so by hooking monomers of nucleic acids together, we can make a whole strand of RNA or a whole strand of DNA. And again, this will come up later on when we talk about genetics and we talk about protein synthesis. But nucleic acids are important for coding the information that results in the genetics of life, if you think about it that way later on. So for example, later on we'll talk about a person's skin color or eye color or hair color, and how those particular traits that a person has that you can physically see are related to genetics. And then the genetics, if you break that down, are going to be related to the sequence that we put these nucleic acids in order. So that's it for now. That's a good start for us to dive in more into our macromolecules, and we'll continue on from there. Everybody have a good day.